Okay. Yeah, so there was another speaker who had questions from the audience regarding what's the right distance metric to use on sort of big biomedical or other scientific data. And rather than directly answering that question, I thought that after listening to my talk, you guys might get a better idea of what that is. So I just wanted to put that in there. So kind of trying to loosely connect between talks. Um, so some of the motivation for my work does come from very high dimensional data. Oftentimes, these can be characterized as high dimensional point clouds. So, for example, single cell RNA sequencing is something like 20,000 dimensional, and it can have, thousands to millions of data points. You get the same thing with molecular data. And maybe later in the talk, I'll talk about how you can regard molecular data also as point clouds. There's a variety of data types that have always existed, but now they're stored more. For example, fMRI data is stored in large databases like Abide and Brain Allen Institute, Brain Atlas type databases. And patient data, of course, is ubiquitously stored in EHR and things like that. So these bring us a lot of sources of high dimensional data. The question is, how can we extract any kind of biomedical or scientific insights from this kind of data, given that it's hard to basically look at it and intuit it in any way? First off, most of these data types, particularly in the biomedical sciences, have a lot of noise. So one of the issues is to remove the noise and retain some kind of signal. Another thing that's very difficult is because of this very, very high dimensionality, it's hard to tell what the structure of the data is and what the shape is. And I understand you guys have learned about topological data analysis, and so that's very much in play. But what you might realize as I go on is in computational topology as opposed to pure topology, it is actually all based on distances and some kind of geometry of the data. And that's kind of what I'll be bringing you. What is the geometry and how should we think about the data? And there's also many modalities of data, which I'll talk about less. And then final challenge in most of these data types is that you don't have continuous measurements. You have static snapshot measurements of some type of a very continuous phenomenon. And sometimes you have continuous measurements at the population level. So for example, if I went outside and took blood samples of 100 people to see what the state of COVID is on campus, and I did the same thing next year, I won't have the same students, but the blood samples will still give me an idea of what is going on with the COVID dynamics. That's the idea of learning dynamics from static samples. So these are all sort of fundamental challenges, but I have found in my work that a really key assumption that actually helps address these challenges is the realization that scientific data is often sampled from a manifold. So it might look like you have points, but underneath the points is some kind of manifold structure. Of course, it's often true that things are splayed out of the manifold because of noise. But then you see what the solution is. The solution is to learn the main dimensions of the manifold and restore it, right? So once you have this manifold as a scaffold, you'll start to understand how you can deal with some of the challenges I mentioned here, like, for example, denoising data. So let me just step back a second. I don't know what the various backgrounds of the students of this workshop are, but what is a manifold? And without getting too deep into like a differential geometry class, I would say it's a topological space that locally resembles a Euclidean space at each point. So one answer to the question that I got in the Slack was, how do you measure distances? Locally, you could measure Euclidean distance, but globally, that's not going to be a valid measure because of the curvature of the manifold. A Riemannian manifold is also a useful model to have in your mind, and that's a smooth manifold. I'll talk a little bit about why should our manifolds be smooth or manifoldy at all. And in a Riemannian manifold, mathematically speaking, it has an inner product defined on the tangent space, which gives a notion of a local metric or an infinitesimal distance. And so if you use this idea, it means that if you've sampled a lot of data points along this, you can somehow measure Euclidean distance locally, and then you can learn globally what's going on, what is the shape of the manifold. And this is the approach we take. And once you take this approach, you can learn a lot about the manifold, which will help you understand challenges. So let's get back to this idea of why should scientific data be generally manifold structured? It's more often manifold structured than it's not. And the reason is because when you measure different features, and let's go with the example of measuring many dimensions in cellular data. Each of these dimensions is not going to be uncorrelated like on the left. The reason is because you're measuring different aspects of the same entity. It's kind of like if you're measuring the position of your left foot, it's got to be really related to the position of your right foot. It cannot be, outside in a field somewhere, because you're measuring aspects of the same entity. And so because we're often measuring interacting aspects of things like cells, there has to be a large amount, I'll say correlation, but more precisely, mutual information that restrains the state space of this. And then so this gives you the lower dimensional aspect of it. And now the idea that this is somehow smooth, it's because because you have to remember, for example, biological systems and biomedical systems have an underlying generator. It's the evolution of this entity, like a cell differentiating or a person progressing through disease. And these types of progressions happen gradually. So if you have enough samples, you'll get the progression as it's happening gradually. So smoothness is a fairly safe assumption as well. So that's a little bit of kind of why, logically speaking, these kinds of entities would have to follow manifold structure. So then if I've kind of hand-wavingly convinced you that this could be a useful thing to do, let's see a little bit about how you actually do it. And this brings us to the topic of this workshop is, learning about graphs. So you can learn about underlying manifolds from sampled data by converting them into basically a weighted, potentially fully connected graph. And a very, very typical way of taking point cloud data and turning them into graphs is this process that I'm showing in this slide here. The original data is observations by features, and I'm harking back to my scientific data by calling observations cells, and the columns, which are the features, genes. Okay, so we do compute Euclidean distances on these, but then we localize the Euclidean distances based on the manifold assumption with a kernel. And you can think of it as a Gaussian kernel, but often we use an adaptive kernel and a kernel with other features. What this does is localizes this connectivity. So if I thresholded this kernel right here, for example, you might have a graph like this where only the local relationships are preserved. And now in order to get global relationships, what you have to do is you have to reconnect, but through the paths that are afforded by this graph. And this is really what I'm talking about when I'm talking about diffusion. Diffusion is a way of using these local relationships to get global distances and global connectivity through this graph, which is really like a mesh on the manifold of the data, if that makes sense. Oh, I see. Okay, so in order to reestablish global connectivity from local connectivity, one of the things we do is we Markov normalize the matrix. That means we row normalize it. So you take each row in the adjacency matrix and divide by the row sum, and this gives you a normalized matrix. And in particular, when you power this matrix, you get t-step random walks on the graph that I showed you. And you'll see the connectivity of this is very clean. If you look at the distances via t-step random walks, they're all totally on the manifold. So this is a very good way of learning, actually, the manifold. The idea of the diffusion operator actually came, almost, 20 years ago now, from actually my colleague, Rafi Koifman at Yale. Back then, they specifically used it as an intermediary computation to getting a diffusion map. So diffusion map is actually the eigen decomposition of the diffusion operator. And because this is a Markov matrix, its eigenvalues range from 1 down to 0, and its eigenvectors, for example, give you what's called a diffusion map representation, where points in your original data space, for example, the second data point, its new embedded coordinates are given by these eigenvector entries, weighted by the eigenvalues. And this is called the diffusion map representation. And here you have a distance called a manifold distance. And this is a pretty good distance between these entities, but actually you can use the diffusion operator in other ways. For example, in information geometric ways, like you just heard about. And I'll talk about that in a second to get yet other notions of manifold distances. So while the original way that diffusion operators were defined were as intermediaries in order to get a diffusion map, we actually use them in a lot of different ways where we don't ever eigendecompose them. So one of the ways is to create signal processing filters. And this is based on the realization that these eigenvectors actually form frequency harmonics on your graph. So these are the same eigenvectors you'd have of the random walk normalized Laplacian. And so by analogy, you have the same eigenvectors, flipped eigenvalues. So the higher valued eigenvalues are the lower frequency harmonics. So if you have a grid graph like this, this second eigenvector, which has the second largest eigenvalue, will give you a very slow moving trend on the data. And higher eigenvectors give faster frequency trends on the data. And now you can see that if you had noise in your data, you could actually perform a low pass filter, as in signal processing. And this would give you a way of restoring data back to the main dimensions of the manifold and away from these dimensions. So you can do a lot with these manifolds. So one types of things are signal processing transforms, which I'll talk about a little bit, and how that leads to a sort of topology. Another type of thing you can do, also related to signal processing, is optimal transport. So if you're trying to measure two cellular populations from the same system, for example, let's say you had a diseased individual and a healthy individual, you can model their joint manifold and look at how their density differs or how far they are from each other. And this is lifting the manifold distance up into a distance between entire point clouds of data. You can, diffusion geometry can also provide a scaffold for topological data analysis, which I'll be talking about in a little bit. And finally, you can also use them as regularizations within deep learning. So I'll be covering a few of these topics as they've occurred in various papers that we've published. Feel free to ask more questions here or on Slack. But first, I'm just actually going to start with PHATE, which some of you guys might have heard about. But one of the reasons I'm talking about PHATE here is to show you what you can learn from diffusion geometry. That might be a little bit different than diffusion maps, but still gives you a notion of a manifold distance. And this is a distance that can be preserved in low dimensions. And that's really the key to it. So one of the most important things we want from scientific data is really to understand what's happening to the underlying entities. What's their structure? Do they have groupings? Do they have trajectories? This kind of thing. And one of the key ways that scientists do this by visualization. And what we realized when we were visualizing our own scientific data is that most visualization methods are not focused on preserving data geometry. And there's no sort of magic behind it. If you want to preserve the geometry, you have to put it in the algorithm. And if you look at these other algorithms, the designers that's not what they were putting in. So for example, TSNE and UMAP are popular visualizations. TSNE also came out around 2008. TSNE is in stochastic neighbor embedding. And so the penalties that were designed and put in there are to preserve neighbors. So if you're trying to preserve just neighbors, unless your manifold is uniform and perfectly connected, you're probably not going to be preserving the global geometry. And we see this on this artificial tree data set that TSNE and UMAP can create shattering, because overall their goal is to preserve neighborhoods. PCA, on the other hand, is a linear method that projects your data down to 2 dimensions. So in particular, it does not have the ability to denoise in these nonlinear directions the way that the nonlinear diffusion operator eigenvectors do. And so you see sort of a linear projection with a lot of overlapping stuff. Diffusion maps themselves, they of course contain all this information, but they contain it in high dimensions. So they are very good for clustering because they'll split out different trajectories into different dimensions. So if you're visualizing 2 dimensions, on average, you get 2 trajectories. So in order to see all the trajectories, you have to look at all the other dimensions. So what we wanted to do was collect the same information that's in a diffusion map, but in a low dimension. And that's kind of motivating PHATE. And so PHATE very much uses the process I talked about. It turns point cloud data into graphs from going from distances, localizing them into affinities. Here we use a special kernel called an alpha decay kernel, which you can read about in the paper. But we power this Markov normalized operator created from the affinity to a power t. And this gives us t-step random walk probabilities. And now between these probabilities, we take an information theoretic distance or a divergence between them. And this creates a distance matrix again. And this distance matrix can be embedded with one of the varieties of fancy MDS, for example, metric MDS. And that's a snapshot of how PHATE works. So just to tell you why we need every step. So the distance matrix is necessary to get the original distances. But of course, you can't rely on these distances when they cut across the manifold because they won't be realistic for your system. So for example, if these were cells, they differentiate like this. So these cells are actually quite far from these, and that's not reflected in the distance matrix. So you have to go to the affinity matrix. The affinity matrix localizes it, but it's still pretty noisy. It's really the powered Markov matrix that cleans these affinities. And so this is one of the reasons we computed. This is the idea of denoising and tackling the noise. So now what we've done is we've globally contextualized every data point. Every data point is described as a t-step random walk to every other data point. And this is a way of globally contextualizing, but it's also a way of deriving a statistical manifold. Now each point here is a probability distribution. And so if you have each point being a probability distribution, you can take a variety of distances, the kind that you take between probability distributions. And for computational sake, we started with an M divergence between them. Currently, we're actually working on a Fisher information metric between them, but I'll have to talk to you guys offline or when I'm further in that work. So the potential distance is actually taking the log transform of these probabilities and taking the Euclidean distance of these log transforms. And we relate it to the diffusion distance like this. And what you'll see quickly is the potential distance is sort of a triple on the diffusion distance, because what it's doing is it's looking at these diffusion eigenvectors and summing over all other vertices. And you're getting these diffusion inner products here. So some of the features of the potential distance are that it damps diffusion probabilities with a log factor. So that means it increases the importance of far points as much as near points. Of course, it sums over diffusion inner product. So you're looking at what's the probability of going from this data point to all others and this to all others instead of direct distance between those 2 data points. And it accounts, so it basically accounts for how correlated are random walks starting from each data point. So it follows the manifold structure. So with this potential distance, we collect them into 2D using metric MDS. And so you get this kind of structure. So this is another branching tree structure we drew. And here is PHATE. But what you see is each of the diffusion eigenvectors is highlighting one branch here. So they can actually work quite nicely with PHATE visualizing the whole structure and diffusion operators picking out individual branches for further study, or ordering the individual branches even. So getting back to how we would use this in a scientific context. So the first system that we tried and we're still working on is embryonic stem cell development. This is a differentiating process where you have human embryonic stem cells, so actually the kind of cells that we all originate as, and they're grown in this kind of 3D sphere culture, which gives them kind of this kind of context, and they start to differentiate. And they quite naturally differentiate into these different lineages, like cardiac or muscle lineages, neuronal, neural progenitor, etc. And so what you see here is these human embryonic stem cells, and I'm telling you this because I've know this from the biology, is that these are the human embryonic stem cells, and they start to differentiate into endoderm and ectoderm, and then these start to differentiate further. So especially for biologists who has an understanding of this system, they can project features back onto it and see how they're evolving with these lineages and things like that. And you see the other visualizations don't keep the structure intact, so you can't do quite the same study. And you can see here is an area of sparsity. And this kind of area of sparsity is typically where something like t-SNE just shatters the plot. So we sought again to kind of quantify what it is FATE is preserving. And since it seems useful for scientific data, it stands to reason that that could be potentially having to do with what it preserves. So our hypothesis is that it preserves denoise manifold affinities. And we propose this as a metric also in that paper for evaluating other visualizations that may be coming into the scientific domain. And the idea is you can take a noiseless data simulation, and in this one, geodesic distances are actually manifold distances. And so if you measure geodesic distances versus embedding distances, what do you get? So this is geodesic distance on the noiseless manifold, and then we add noise and we use embedding methods on it. And it seems that this is where FATE achieves improvements over other visualization techniques. It has to be mentioned that we found that FATE improves also adjusted RAND index, which means the global positioning of clusters. But for the purposes of this talk, I'll focus on this manifold affinity preservation, which means that what FATE is really doing is within the cellular manifold, how far are things from one another. So this distance can be used as well. So recently we've used it on other types of data. For example, visualizing the loss landscapes of neural networks, which was published recently at the Intelligent Data Analysis. And what we actually see is if you go to a minima that your neural network reaches, we do what's called jump and retrain sampling. And when we do the jump and retrain sampling, we see a very different type of behavior. When you look at this minima here, jumping out and coming back, you see this kind of flower petal pattern. And this is in minimas that generalize well. Minimas that have poor generalizable capability, they tend to not have this pattern, and you can have random walks that escape or kind of meander before they come back. And if you're wondering, oh, could I have done this with like, for example, other things? And this is actually the total persistence. So because it gives us these nice loopy structures, we actually quantified this with total persistence and classified it. So if you're looking at other visualizations, it would be very difficult to get this insight from that, because as with branching structure, trajectory structure could also be shattered pretty easily. And so that's a little bit of how we use the diffusion geometry to get a visualization. So now let's talk a little bit about topology, particularly computational topology. So as you guys probably all know, topology allows you to look at entities at multiple levels of resolution. This is also extremely important in biology because biological entities have multiple levels of, or even fractal levels of organization, and there's a high degree of complexity here. And of course, in topological data analysis, we quantify these properties like Betti numbers or Euler characteristics, or these types of things that you have probably heard about. But if I show you how Vietoris RIPS works, for example. So we have a point cloud of data, and what we're actually doing is we're growing epsilon balls. So there's a clear notion of geometry and distance here. So this point is further than epsilon away from this. So there's a distance, right? And so computational topology, of course, scans through these distances, and it comes up with these features, birth-death features of these. Here we have connected components, and we have these cycles that emerge, and of course you can see when persistent features emerge, and this gives you a characterization of the data. But as you saw before, if you use ambient distances to do topology on your data, you're not going to get something very meaningful, because remember the new distance I had to come up with, going from distances to affinities to diffusion probabilities to statistical manifold. So naturally, we feel that the distance that you'd be using and the scaffold that you'd be using to do topology would be improved if you were using the manifold structure that we just learned. And this is exactly what we've been developing. And in fact, together with just not just the scaffold or the graph of the manifold, we've also proposed a new type of filtration. And this was first proposed in this IEEE Big Data paper. Now we have a preprint that's is under review at SciMods, the SciAm Mathematics of Data Science. And this is the way this filtration works. This filtration actually uses the diffusion operator itself as a filter back on the data. So what's happening here is you get your diffusion matrix. So if you applied it to your original data, what happens is you condense the data points because they're replaced by the average of their diffusion neighbors. And this is what and this is a type of filtering. I'll talk about it in a second. This is a type of filtering in many ways, in topology as well as signal processing. So you see the different iterations of this condense your data points into larger and larger groupings. The advantages of this is particularly when you raise your Markov matrix to a high power, you're getting this condensation happening on the manifold of the data. Another way of thinking about this is that this light one, the cells are nodes on your graph, and the genes are mRNA counts on this graph, and then you're taking a graph Fourier transform, and you're coming up with the random walk normalized Laplacian, and what you're doing is you're taking the eigenvectors of this, and you're loading the eigenvalues, and you're doing the reverse operation back. So the reason we didn't have to do the eigen decomposition is the powering actually produces a filter shape that's like a low-pass filter right here. So you can look at this in the frequency domain, what's happening when you apply the powered diffusion operator. In the frequency domain, or in this graph Fourier domain, what you're seeing is as you raise your power of t, you're getting sharper and sharper low-pass behavior. And so previously, we had used just one step of this to create an algorithm called MAGIC that denoised data. But now we're using it over and over again and recreating the operator to create a diffusion filtration. So if you use it one time, what happens is you kill a little bit of variability. When you kill a little bit of variability, you restore data back onto the manifold. So if you're looking at data, trends in the data, they reform. Whereas here, I added actually a drastic amount of noise. 80% of the values were set to 0 in the cellular data. And then it was restored back here. Now, when you do it over and over again, what you can get is hierarchical organization of your entity. So the first place that this was applied together with our collaborators was in this Nature paper that was published last year. So we were looking at the connectivity between neurons, and we created an adjacency matrix based on physical connectivity. And then we applied this condensation technique to it. And this is actually all of the neurons in this worm. So this gives you a picture of how all of the neurons act in concert with one another. And what this allowed our collaborators to do was understand how different neuronal circuits had been characterized, both communicate with each other, and perhaps additional neurons that hadn't thought to be part of those circuitry was added into it. But I'm not an expert on neural connectivity, but I just wanted to mention how you would kind of use it. So more recently, we've been studying the properties of this kind of condensation filtration. And we have been able to prove that eventually, every time you run the condensation, you'll converge to a single point. So that means you'll sweep all the way from every point being itself all the way to your entire data being one point. And given the right epsilon schedule, you will not get stuck. So the proof idea is as follows. Basically, what happens is, if you think about the convex hull of the data, the diffusion operator strictly pulls things inside the convex hull. So that means your convex hull shrinks, and this keeps happening, and it happens at a rate proportional to the smallest value in your diffusion kernel. And so this gives you an idea of what that parameter selection should also be. We also had a spectral proof. It's a little bit more difficult to explain the spectral proof. When the diffusion operator is applied to a function, the first non-trivial eigenvalue is a bound on the resultant magnitude. And so again, we can select the bandwidth of the diffusion kernel such that the feeler value goes away from 1. And what's more, we can speed it up now. Every time it's going towards 1, we can change the bandwidth, and we can make this speed up, and this leads to convergence as well. And so recently we've combined the condensation process with the PHATE process to come up with a multi-scale visualization that allows you to zoom in. So if you look at that same kind of data, and this actually appeared last month, I think this is an old slide, so it appeared in May. And it was actually on the cover too, because our graphic designer made a very cool cover. And so you take this data and you're thinking, I want more clarity on what's happening here. So you can kind of, first off, you get a natural clustering of the data. You can zoom in and see subclusters. And this can give you a natural way of kind of interacting and coarse graining with your data. So now I want to talk a little bit not just about these points lying on a manifold, but what if you had multiple data sets that were on a common manifold, and what could you do with sets of sets like this? So each data set is a set of data points, and does diffusion geometry tell you anything about how to compare these diffusion data sets? So the first effort we had in this vein, it was published last year. It's a method called MELD. And what this method really does is it estimates density in density over the manifold using graph signal processing. And then it's used to compare compositions of two different data sets like this. And let me motivate this. So why would you ever want to do this? So these days, you don't just collect one single cell data set or one cohort. You usually have a control and a perturbed sample, or healthy and a disease. So if you want to compare these conditions, given how complex this data is, it's not clear what's going on. So you could try to say some kind of average is different, but that doesn't really capture the picture of what's happening. So here you have some kind of stimulated and unstimulated cells. There's quite a lot of overlap. There's some regions where they're not overlapping. And overall there's a massive amount of noise. And so what we actually want is sort of a scoring or a coloring on the manifold that says which sample is enriched where. And so the picture that we have in our head for our goal here is we want to estimate density on the manifold of 1. So let's say this is the manifold or the cellular state space. We have the density of one sample and the density of the other sample. And this allows us to differentiate between these densities. And what you can do after you get this difference is you can do a difference aware clustering. So in traditional analysis, what people have been doing instead of this is just clustering the data. So if you just cluster the data, there's no reason why your clusters have anything to do with the resolution of the changes in your manifold. So you might get spurious results of cell types that you think are up, but it's not actually that cell type that's up, it's some small subset. And so we think that you should postpone the clustering until you've learned the manifold of your data and understood density on the manifold. And so in order to do this, we established a kernel density estimation method that's very sensitive to the geometry of our manifold and the waves we've sampled it. So what we do is we combine the two data sets into a common data graph. And once we get the common data graph, you can create indicator signals for which sample each point originated from. So you see you have this 1, 0, and here it's just 0, 1, it's the opposite. And then we create some kind of a filter, again, a graph Fourier filter, to create a density estimate. And then we can just compare these density estimates in some way. Okay, so just to give you the intuition of how to create this filter, you can create it directly using an optimization. So one of the key features you want from a density estimate that's different than just using histograms is you want smoothness, because you believe that it's resistant to noise in the actual sampling the way a histogram is. So what we do is we just take a graph Laplacian of the data, same Laplacian I showed before, and you can require that your signal, respects the indicator when it can, but it's also very smooth on the manifold. So this way, it respects the histogram, which is your indicator, and it's smooth on the manifold. And this one you can actually analytically solve, and it gives you a spectral filter that you can use with Chebyshev polynomials. But what we actually found was that this was a little bit still numerically unstable and inaccurate to some extent. So we started to use a different Laplacian. We started to use a heat Laplacian, which we define like this. Now we reformulate it in this way, and this, because this differential equation is a unique solution to the Fourier heat equation. It's much more stable, it's been much more studied, and the coefficient, and so you get coefficients that are between 0 and 1 at every vertex. So when we use this, you can find which parts of the manifold are enriched for control condition versus experimental condition. And actually, you can use even the frequencies of these signals to come up with much more informed signals. And actually, we defined another likelihood over this, where we can actually come up with a score for each cell about whether it's more likely to come from the control condition or the experimental condition. And so you could just threshold this likelihood, and this would give you areas of the manifold that are enriched for one condition or another. So that was comparing two conditions. And if you look back at this T-cell stimulation experiment, you have this kind of mixed cell state. Here we have this likelihood score coloring. And when you do the likelihood score coloring, you can actually threshold to cell types that are very different between the two manifolds, and then you can study what's happening. And when you do that, when you take the cells that have very different likelihood scores, and we looked at what genes were expressed in it, we can actually see differential expression exactly of T-cell stimulation genes, as opposed to other ways of clustering, many other ways of clustering without the signal, where you don't nearly get that sharp of a signal, because each of these gets a significance score if you do gene set enrichment analysis, and significance scores are much less for other ways of doing it. So now that I've talked to you about this a little bit, so we can kind of see how these processes can interact with one another to find different kinds of biomarkers and things like that. So we actually have been studying a variety of diseases with this, and one of the diseases is age-related macular degeneration. So our collaborators generated their first single-cell atlas of AMD, where they sequenced 17 retina, 7 from healthy, and 4 from early, and 6 from late stage of this disease, which can cause blindness. So PHATE shows you the overall structure of this, and you can actually see using the condensation homology that the most persistent clusters are sort of the ones that you think about, the canonical data types that the biologists think about. But when you look at how differentially enriched they are between the healthy and the AMD, what you start seeing is this very strong immunological signature with microglia and astrocytes highly differentially expressed. So now we zoom in on our visualization and go into a finer granularity of visualization, and we've colored with MELD again. And when we color with MELD again, we see there's a subcluster that's responsible for sort of driving the early AMD. And the very interesting thing we found was when you look just at the signatures in that subclusters, we see it replicated in Alzheimer's disease and multiple sclerosis, which are other neurodegenerative diseases. This could be potentially a way of deriving an early stage biomarker that's pan-neurodegenerative disease. Later on, I will say this kind of common signature disappears and these diseases become more individualistic. And these signatures can be validated in a variety of ways, including imaging technologies. Let's see. Sorry. Yeah. I'm just trying to see how much time I have. If I can go into another disease case. Since we're all dealing with the pandemic, I can talk a little bit about COVID. We also received a lot of single-cell COVID data from patients admitted to Yale New Haven Hospital. And there were a total of 80 million cells measured from 4 different flow cytometry panels. And so one of the panels we visualized on all of the patients that were enrolled in this study. And here we have a certain granularity that we're visualizing with multiscale PHATE, and we've colored it by the MELD likelihood scores. And this gives you a very particular part of the manifold that's enriched in patients who had adverse outcome. That means they died or went to hospice. And so again, when you zoom in and look at this, you see very specific. So here you can attribute it to specific cell types that have a higher mortality likelihood. When we looked at our T cell, we got a kind of an interesting story too. The T cell panel, it also shows specific enrichment. When you zoom in, you see very specific non-canonical types of T cells conferring mortality likelihood. And sometimes the trend can kind of be the opposite. So overall, T cells seem protective, but when you look at this subset of T cells, suddenly the mortality likelihood increases. And then when we ran this through random forest classifier, we can get importance of these different features that we found at different levels of granularity, leading to a little bit of predictive insight. So that's a way we measured basically differences between just 2 conditions, and we were able to get a lot of biological insight on some diseases from just measuring difference between 2 conditions. More recently, we've talked about measuring differences between multiple conditions. So for example, we have those 200 patients. Can we measure the distances between every pair of patients? And that's a lot of optimal transport to run. And Euclidean optimal transport might not give you the right kind of distance. So we've defined a diffusion earth mover's distance published in ICML last year. Earth mover's distance, or Wasserstein distance, is a 1 Wasserstein distance, and it basically measures the amount of work to go from one distribution to another. Typically speaking, you develop a transport plan, which is a joint probability distribution, and you penalize it by ground cost. And you take the pointwise multiplication of this summed up as the cost, and you run a linear program like this, where the marginals have to agree with the 2 original distributions. An alternative to doing this is to produce a so-called witness function. The important thing about the witness function is it has to be a 1 Lipschitz function that exposes the difference between these 2 probability distributions. And this is a result of the Cantor-Rubinstein duality that shows that in this witness function, the expectation is very different under the 2 different distributions, but it has to be 1 Lipschitz. This doesn't necessarily give a transport plan, but it's actually a pretty fast way of getting a distance in many ways. And I'm going to give you an example. This is work from 2008, where what basically the intuition is that you take histograms of your 2 different conditions, and you take the difference between those histograms. That could be a witness function, but there's no reason it has to be 1 Lipschitz. So to keep it smooth and varying smoothly, they wavelet decompose it. And if you limit the scales of your wavelets, you do get the smooth varying function. But this was based on original wavelets, which are defined only on image signals, and these histograms are defined on a 2D grid. But we have extremely high dimensional data, not 2-dimensional data. So instead, what we're doing is we're using the same trick as in MELD. We create a common data graph, many indicator vectors, and instead of a multiscale wavelet transform, we use a multiscale diffusion filter based on the MELD filter, and we create a multiscale histogram. And now an L1 distance between these multiscale histograms of these distributions, or multiscale diffusions of these different distributions you had, actually creates a pairwise Wasserstein distance matrix. Now that you have pairwise Wasserstein distance, you can embed all of these people. And now you get a people embedding instead of a cellular embedding. And so this ground distance is actually a manifold distance because it's based on diffusions on your data manifold graph. And so this is a multi-scale diffusion distance that's leading to where you take the multi-scale diffusion distance between the different histograms. And when you sum these different scales up from K0 to KK, and remember these scales can be limited, so they're usually dyadic scales, you get a Wasserstein distance. And we've shown in the paper that this is the discretization of a continuous result from Lieb et al. That shows that this is a Wasserstein distance. So the thing we get out of it is that we get the speed of some of these dual forms, but we get much more of the accuracy of a primal form in terms of P10 neighbors and Spearman correlation with ground truth distances. And when you look at those COVID patients, again, using this, the diffusion EMD most organizes them as our feature set did, with patients who died here, patients who survived here, and everybody in between. See, these other distances don't quite organize them quite the same way. So that's how you use multi-scale diffusion, not just a single scale, and multi-scale condensation to get a lot of features, topology, and distances out from this. The last thing I'm going to talk about is how you can use these in learning. So for this, we use a diffusion-based framework that we've been developing called geometric scattering. The idea, our first idea for this, or first motivation for this, was that we wanted to find a representation for molecular graphs where global connectivity is important, which is something that actually graph neural networks are pretty bad at describing, where the internal representation we get, for example, inside an autoencoder, is faithful to graph edit distance as well as some kind of property. So for example, binding affinity. So we want this space to be dual organized by a property as well as the graph distance. And we want it to be invertible so we can generate new molecules out. So molecules also have a manifold structure, but they're usually sampled in certain ways where you can convert them into a graph. Sometimes you can convert them into a mesh graph. Sometimes you can convert them into graph just based on atoms or amino acids. And if you just put the atom through a graph neural network, one of the problems is it uses a lot of local aggregations, which of course can't distinguish between certain global constructions. So instead, we want to be able to distinguish between these global connectivities. So in a molecule, if it had an extra loop like this, that would be very different than this. So if you look at local connectivity, most of the signals would have the same one, but if we're looking at T diffusion steps and comparing it to T-1 diffusion steps, you can already distinguish between these two graphs because of the nodes that you can get to in one step versus the nodes you can get to in two steps. So for example, this node has very different diffusion probabilities. So the idea here is to use the same kind of diffusion operator. And if you had a molecular graph that just came from the atoms, you could turn it into a diffusion operator just by adding laziness and normalizing it like this. And so this lazy random walk operator, you can power it to two different scales. And this creates what's called a diffusion wavelet. And we use neural networks with diffusion wavelet layers because remember, this is just matrix multiplication on these matrices, no eigendecomposition, just like graph neural network Laplacians. And we created a learnable version of this that was published in Machine Learning for Signal Processing last year. This learnable version learns the scales, learns the extent of laziness, and it contains a diffusion module. And this is diffusion module performs diffusion. And then you have a aggregation module that compares different scales of diffusion, and a scattering module, which takes coefficients of your signal with multiple scales of wavelets. And so scattering transforms are sometimes described like this. You have a signal, you have different scales of wavelets, you get coefficients out, and then you do it again. You can apply these same scales of wavelets on your coefficients, which gives you a next order. And so we created an autoencoder that uses these layers, and it's penalized to predict a property. And so what we saw was that this kind of autoencoder if you took a graph and you just mutated it one vertex at a time, will give you this kind of trajectory structure and latent space in a way that others don't. And then when we put in molecules, we can see, for example, two different conformations of low energy of this bistable molecule that we knew was bistable by construction. So now when we embed sets of molecules there, what we get is a latent space that's smooth with respect to structure. So you can start from somewhere and you can preserve as much of the structure as you want and get it to a state that you like better with lower energy just by traversing this manifold, and you're generating new data points out. So right now we're doing this on molecular data, and the only addition we've done is we have an adversarial generator instead of just the autoencoder or decoder. And so these are different types of molecules that it can generate, which could be, it can either be small molecules, proteins, or anything where you can get kind of a graphical structure, and we're doing that a little bit. So this is how the network looks. It uses the learnable scattering module, uses regression network to predict the penalty, uses the generator to generate out. It has a decoder just for the autoencoding penalty. This gives us the highly structured latent space here. And here's some zinc database molecular results on this, and we have more. I had to update this slide. So just in conclusion, as I'm starting to wrap up, the main takeaway message was basically that a lot of scientific data pretty naturally lies in a collection of manifolds, which are these low-dimensional, locally Euclidean structures. You can take advantage of that by how you create the graph and then what you do with the graph. For example, turning the graph into a diffusion operator, looking at its eigenvectors and things like that. And these help learn a lot about the underlying entity in many ways, so it gives us sort of a powerful tool set. But are we done? No, there's a lot of work left to be done. There's a lot of open questions. I would say like the kind of like the golden unicorn that you want to find here is you really want to learn, you know, how these biochemical systems work, but not at the level of detail that they already are at a level of detail where they're interpretable, but potentially dynamic and simulatable. So this creates a big inverse problem. Some of the stuff that we're working on now has to do, for example, one of the projects that we're working actively on, and we had a paper last year and we submitted one this year, is on learning dynamics from these kinds of static snapshot data. The manifold assumption helps there too. And this can give us certain kinds of causal inferences that we wouldn't have had before. We're also trying to learn dynamics on a graph scaffold because you can also represent regulatory networks inside cells as a graph. So this is maybe something for you guys to think about too. So can we learn these kinds of dynamics on a graph? Because each cell can actually be represented as a graph, much like each molecule can. Another thing we've been touching upon, and I saw the last speaker doing it, so I wanted to make sure I mention this, is you can also, we're also trying to quantify curvature in this kind of high dimensional data. The idea of curvature being important is that there is a lot of negative curvature in biology, and that corresponds to branching structure, actually, the tree-like branching structure that you saw. And so because the diffusion operator is closely related to the Laplace-Beltrami operator, we are able to use diffusion, and we've proposed a diffusion-based curvature method that looks at the laziness of a random walk, assuming an anisotropic kernel that weeds out for geometry. And so we have a preprint on this if you're interested. It's looking within a specific radius and looking at the laziness of a random walk operator on that. And we're seeing some good results where you have negative curvature inside the donut, positive curvature outside, flat at the top, all the usual stuff. And we actually hark back to Bishop Gromov in order to prove this. But this is not an ideal metric because it always gives positive values. We would love to get negative values here. And actually, we would want this to be actually a curvature tensor. But these are more open questions that remain to be handled. And another final sort of challenge is can you get neural networks to learn structures? So we've showed a couple of cases where we regularized neural networks to have certain kinds of geometric structures. The neural networks are still sort of in Euclidean space. Can they learn the sort of more natural space that these things have to go into? So these are some open challenges. We have a lot of this flavor of work in our lab if you want to read it, and all the code I talked about is on our lab GitHub. So I'm just gonna end there and see if we have questions.